Thank you. That was quite a wonderful introduction. I'm really touched. Yes, it's really nice to be here today. Wonderful. I was really happy to get the invite from Kia, who has long been a mentor of mine while she was at Oxford. We're so sad to see her go, but it's wonderful to see, Kia, where you've landed. This seems like a really special place, and I'm sure it's only going to get more so. I love the idea of asking people to talk about their background, so much so that I actually do it quite a lot. So this was not a big ask. I have slides. I already had slides. Like talking about myself, I guess. Right. So yes. So I grew up in this country, in Iowa. When I give this talk in the UK, I always love to make people guess where Iowa is. And there's like one person who'll get it right. And everybody else pretends. Anyway, it's there. That's Iowa. I started off at the University of Minnesota, and I was initially a creative writing major, but quite quickly decided to move into psychology. And I remained as a psychology major even when I transferred to the University of Illinois. But I actually quite early on was frustrated in the cognitive psychology courses that I was taking at what felt like very crude ways of interrogating cognition with the tools that we had back then in the mid-90s. And I transferred to computer science actually because of seeing in a textbook, which they must have really quite quickly adapted this into a textbook, given the timing of all of this, seeing in a textbook functional MRI and thinking, that is the thing that you want. I'm tired of people talking about, testing reaction times and things like that. These very crude external measures. This intrinsic thing seemed really to be what we needed, but also it was such a new technology that I thought actually a lot of the major leaps in this space are going to be in the technology itself. And so I decided I wanted to get on top of technology, and so I became a computer science major. So already I'd kind of shifted my interests, and I quickly added this slide. When having been taken on a tour earlier, I got to see that you have the technology that I worked with coming at least. This is what I did when I was at the University of Illinois. My first research was working with a cave virtual environment, and this was really cutting edge stuff at the time. And you can see you got to wear really cool glasses and really bad clothes. And this is what I worked on, is a program called Crumbs, where you were supposed to be laying little points in a 3D any kind of biological imaging data in order to make measurements. And so I came up with what was called the silk cursor. It sounded really cool. And I did that. Yeah, that was great. So that was my first research job, was working in virtual environments. And it's really cool to see them coming back from my perspective. So from there, I left the Midwest and went to Stanford and landed in an electrical engineering department. So again, a bit of a shift. It was quite jarring for me, actually, because there was a lot that in electrical engineering, a lot of technical skills that I didn't have. I hadn't had a lot of the mathematical background that was expected, and got my first C, actually, my first year at Stanford, while struggling to get on top of stuff. But it was actually really good for me to be pushed, I think, in a way that I hadn't before. I never really quite struggled with coursework so much as in that first year, but it was a great experience for me because it forced me, particularly, they have a really quite brutal and very notorious qualifying exam that you have to go through to remain as a PhD student. And it forced me to get on top of some of the maths that is very core to what I've subsequently gone on to do. So it was a bit of a struggle initially, but it turned out to be a great move for me. And subsequent to that, well, actually, even before that, there were some lessons that I learned at Stanford that, yes, that's me, little Karla there, looking surly with her lab mates. So some of the lessons that I learned, one of the key things was that I was doing, because I came at it from an interest in the brain, but I ended up in a lab that was a cardiovascular imaging lab. And it was really interesting because they had amazing technologies that they were developing. They were one of the leading labs, cutting edge in what I would now call MR physics, but in developing acquisition technologies. But they were all coming at it from a position of thinking about the heart, and I wanted to think about the brain. And they let me work on the brain, but it meant that I kind of came at it from a very different perspective than if I had been in a lab where everybody else was working on the brain and thinking about it in the traditional way. And so one of the things that I learned is that unusual perspectives encourage innovation. Some of the stuff that I came up with, I look back and I'm really proud of it in the sense that I came up with some very interesting ways of trying to get at fMRI and diffusion MRI that nobody else was really trying to do. But I also learned that innovation really only gets you so far. Because I was in a lab where nobody else was thinking about the brain, I was very, very naive, even about how to look at my own images and really understand what they were telling me. So from there, I then, as a postdoc, moved to Oxford. And at this point, I landed in what was referred to as the MR physics group. So great, I'm a physicist now, right? Which felt very, like, even further away from where my actual training was. And I got a fellowship, I started a group. So again, some lessons learned from those early days in Oxford. I learned that it pays to know your problem. So one of the things that I felt when I came there initially was, I don't know, people are not being quite as wild and creative as I've been during my PhD. But I also very quickly realized that they actually understood much better than I did what the real problems were that needed solving. Not just how to be really creative and come up with wacky ideas, but what's the actual thing that people need a brain image to deliver. And that kind of led me then to sort of finding this sort of happy middle space of understanding what the problems are that the neuroscientists are saying they want solved from people who are a physicist or an engineer, but also maybe working just outside in the mainstream because it allows you to be creative. And it also is potentially where the next big things really happen. And so I think having those two experiences was really valuable for me. It made me into a better scientist from both ends. So this is my group around 2014. This is around the time I got in my second fellowship, being silly at one of our retreats. And around this time, I was starting to move away from the things that I had been trained formally to do. So not just thinking about how to change MRI acquisitions and reconstruction techniques, but thinking about being a bit more adventurous. And in particular, because I'd started to get into the idea of scanning postmortem tissue, I then ended up kind of inevitably sliding into working with microscopy data. And that was completely foreign to me. But it was, again, a really interesting experience because now, this time, I was completely new. I was a novice again, but I'd been through the process before. So here's me learning how to do microscopy initially. You can see that I have no idea what I'm doing, right? I know that there's a task, but I'm not sure what it is. But then occasionally you get a little tiny taste of success and it's like, yes, right? This really is what it felt like, right? But again, like not knowing what the task was, it wasn't so scary the second time through. It's like, I know if I just kind of keep hammering away at it, I'll get there. But I also know that I'm a novice and that I need to acknowledge that, right? Of course, around this time also, I'm just beginning to deal with what everybody deals with at this stage of their career, which is learning how to write grants and learning how to fail and fail and fail. And the fact that even once you succeed, right, then you have to deliver, right? Sorry, I just love these videos. Anyway. So, okay, so yes. So about 2014, I think things were going well. I got tenure, so this was now, I guess, 10 years after finishing my PhD. Shortly after that, I became associate director of the center where Key and I worked together. 2022, excuse me, I ended up taking on an interesting role, which is being the academic lead for EDI and all of medical sciences in Oxford, and that's something I talked about earlier today. It's something that I really value. It's something that I think is really important for all of us to engage with, particularly in the current time, even more in spite of the challenges that we're all facing. Very happy to talk about some of this in the Q&A or in the reception afterwards. And yes, now this year, in about a month's time, I'm going to be taking on as director. So quite a wild ride, at least it was to experience it. But yes, that's kind of where I'm coming at. Okay, I spent a lot of time on that, so we're going to whiz through some science now. So what I'm going to talk to you about today is the methods that we've had for crossing scales, and scales interpreted in a few ways in neuroscience. So most of you, I'm sure, are aware of this sort of way of thinking about what we're doing in neurosciences, which is that structure and function relate to each other in the brain across many orders of magnitude. I've shown about 6 here. I think it's arguably maybe more like 9. But from the perspective of this kind of a diagram, where you see the whole brain far on the right-hand side, I think as an imaging community, we tend to do ourselves a bit of a disservice. Because if you think about what this suggests, it suggests that we as imagers are somehow at the end of a process. It begins on the left and it proceeds toward the right until you finally end up with something at a relatively crude scale of the brain. And I think a better way of thinking about where imaging is in the space of scale and how we address it is maybe to think about it from this perspective, which is that imaging is one of a very few tools that we have in neuroscience that really allows us to bridge and integrate across many levels of investigation. So we can take essentially the same tool and we can use it to interrogate tissue, as I'll talk about today. We can look at causal studies in animals. We can look at living patients. And as I'll talk about today also, we can look at whole populations. And I can't really think of many other things in neuroscience that have that kind of reach, where you can get the same readout in all of these domains. And I think that's a really special thing. So one of the things then that at our center that we have been thinking about is how can we leverage this? How can we really lean into this potential? Because I don't think we do enough of this. We tend to work in silos and not so much integrating across these different domains. So what I'm going to start with is spatial scales and work that we've done linking MRI to histology. And I'm going to focus on diffusion MRI. Now I'm going to make the assumption that most of you know what diffusion MRI is. So this is a form of MRI that allows us to look at pathways and microstructure. So we can look at structural connectivity via mapping the white matter pathways. And we can also look at microstructural properties. So this is sensitive to things that are happening at the cellular level. But one of the challenges that we face, as with all MRI, is a challenge of specificity. So in vivo MRI is notoriously nonspecific. It also has relatively poor resolution. So one of the ways that we can think about how we can relate in vivo MRI or understand in vivo MRI in terms of what it is telling us is to try to relate it to other scales. So for example, to optical imaging, where either by virtue of having imparted some kind of a specific stain or simply by resolving structures, we can begin to have greater specificity than we have with MRI. And so the way that my lab has been approaching this is to think about postmortem MRI as a really important intermediary that can help us to link between in vivo MRI and optical imaging. And it can do that because it shares the same signals as in vivo MRI and the same tissue state as optical imaging. And so by virtue of doing that, what we can do is to better understand what in vivo MRI is telling us, but also it enables us to build on a strength of MRI, which is that unlike optical imaging, where it's very difficult to interrogate a large number of brains or tissue samples, with in vivo MRI we can. And so that's one way that we can leverage the complementary strengths of these different methods in order, for example, to study variability across individuals. So when we first started doing this, it was around the time that a few people, including Tim Darby, were beginning to show how you can acquire postmortem or MRI data in a postmortem brain. And this is an example from a macaque acquired on a rodent scanner. And you can see that it's beautiful looking data. Now, it's quite a lot that Tim had to do to make this work, but we wanted to do human brains. And if you try to do the equivalent, take a human brain and fit it into the smallest scanner you can find with the strongest gradients, which is unfortunately a human scanner, then what you're likely to get is something like this. It's unusable. It's garbage. And the reason is that postmortem tissue, when it's been fixed, there have been a lot of changes, and unfortunately, all of them are bad news for MRI. So around 2010, my lab started thinking about this problem based on an interest from a neurosurgeon who wanted to be able to do tractography in brains that he could then dissect. And after developing a range of techniques that I won't have time to go into today, we ultimately were able to get to the kind of data that I'm showing you here. So this is a 0.5 millimeter resolution whole human brain data set. It took 10 days to acquire, but it's possible to acquire it. We can get those scan times down now. But as you can see on the left there, we're also able to get very sensible looking tractography out of this data. But a lot of technical developments had to go into making this happen. And again, I don't have time to go into that today. We also had to solve other technical challenges. So one of the things that we wanted to be able to do was to take blocks from those brains that we were then going to stain for different proteins of interest, so to do histopathology. And if you're going to do that and you want to be able to make a voxel-wise comparison, you have a very difficult image registration problem. And so for quite a while, we'd kind of gone out and naively just started acquiring this data. It took us a very long time, and you can see the time now, it's 2023 that we finally published this paper, that we finally managed to crack this problem using a series of novel registration techniques, photographs taken of the brain, so that we can now insert a small section into a human brain. This sort of direction is something that we're pursuing a lot in the space of neurodegenerative disease. I'll give you a taste of it, but we're really only just getting to the point where this data is curated and we're able to start working with it. We also ended up getting into developing microscopy techniques, and this is one of the first ones that I ended up working with in order to compare it to diffusion data. So this is polarized light imaging data. It's a number of very standard components to a microscope that you just have to configure in a specific way, and in particular having a series of polarizers, one of which you rotate and acquire a series of images. And when you do that, if you look in the corpus callosum here, what you see is the light intensity is changing as that rotator goes around. And that's because myelin has a property called birefringence. It's basically a polarizer itself. And so you can analyze this data. When you take a whole series of images, you can analyze the data, and because it's driven by myelin, that means that basically when in this rotating cycle the images are bright versus dark depends on the in-plane orientation of fibers. So you can analyze this data and produce beautiful images like this. So this is a human cerebellum where the color is indicating, similar to how we do in diffusion imaging, the color is indicating the orientation. It's just a slightly different color map because we only have the orientations within the plane of the image. But you can see it produces really stunningly beautiful images. And we can compare these to diffusion imaging. So to give you a first taste of how we might try to use these two very different kinds of data to ask questions, this is an example where we're in the pons. Polarized light imaging on the left, and the diffusion tractography at 0.5 millimeter, that same data set I was showing you a moment ago on the right. And what you can see is that in both cases, you can see the interdigitation of different fiber populations where you have fibers that are running vertically in the pons and interdigitating fibers that are crossing between the hemispheres. And the fiber tractography is starting to be able to capture that very fine structure in the postmortem diffusion data as we get down to high spatial resolutions. Okay, so what are we trying to do with this kind of data? There's a lot of things that you can do once you have these kind of data sets together. And I just wanted to give a bit of a flavor of how we're thinking about the value of these data sets. And I want to start with sort of the simpler things and build up to where we can start to do actually some things that are quite a bit more interesting. So the first thing, the most obvious thing, and we are not the first people to do this, this is exactly how people have used this kind of data in the past, is to perform simple correlations. So we have non-specific MRI, and we have some kind of a gold standard based on a microscopy image. And we want to ask, do these two things relate to each other, right? So we can do correlations. Fine. Okay, so here's an example of that very obvious thing that we did. So this is, I mentioned ALS. This is one study that we did in that space looking at the perforant pathway. So this is a pathway in the temporal lobe where it was unclear evidence as to whether or not this might be involved in ALS. And there's reasons to suspect that it might be. I don't have time to go into that. But what this was showing, in this case, we're able to show that there was a correlation in ALS and healthy controls between a diffusion metric, fractional anisotropy, and myelin stain when you acquire this in exactly the same tissue. Okay, so this is a relatively obvious thing to do, but it's a nice demonstration. But we want to do we ultimately want to aim for things that are more ambitious than that. So rather than simply doing correlations, we can set a higher bar for ourselves. We can ask for consensus, right? That means validation. So in other words, if we take some MRI data and we think we have some model as to how that relates to microstructure or how it might predict microscopy, if we take our MRI data, we run it through that model, and we're able to predict the microscopy data, what we've done is actually quite a high bar for demonstrating that that model has some validity. It doesn't have to, it's not enough for it to correlate. It needs to actually be accurate. So here's an example of where we did that. We took that polarized light imaging data. So this is in the corpus callosum. So that's the coronal slice from right through the callosum. You can take a voxel that's about the size that you would have in MRI data. So in the polarized light imaging data, in every pixel at about 4 micron resolution, you have an orientation estimate. So you can take your MRI-sized voxel, and then you can pool those orientations and produce a distribution. And this distribution is something that we try to estimate in diffusion MRI. This is the thing that you estimate in order to proceed to tractography, is a set of orientations within your MRI voxel. And so you can quantify properties from that. For example, a property that is quite common that people try to quantify is dispersion. How incoherent are the fibers in a given voxel? So this is a good target for us to try to demonstrate how you could do this sort of a more of a consensus approach with MRI histology data. And this is showing like one of a few plots that we had in this paper where on the horizontal axis we're showing the diffusion prediction, what the diffusion thinks the dispersion is, and on the vertical axis what the microscopy data is telling us the dispersion is. In this case, it's not actually PLI. We also had some histology data, like classic myelin staining that we used to do the estimate. And what you see is that the blue line is the line of unity, right? So that's perfect agreement. And you can see that actually the measures that we have from diffusion are actually quite accurate in an absolute sense. And that's a much higher bar than simply saying, are these things correlated, right? So the dashed line is the correlation. But we are really not far from the blue line. Now, the thing that really I found quite remarkable about that is that the red points here are in the centrum semiovale, which is an extremely complex bit of white matter where there's lots of different fibers interdigitating. So it's not just in regions where the fibers are relatively coherent that we can get this prediction right. It's in relatively complex bits of fiber architecture, although you see that that's the bit that we get the most wrong, which is not surprising. Okay, so that's an example with a higher bar. But what we think is really exciting is something more like this, where the aim is not to feed one data set into another or to simply compare the two data sets, but to ask, can we co-model the data sets in ways that are meaningful and useful? So what do I mean by co-modeling or joint modeling? Well, here's an example that was done by Amy Howard in my group, along with Saj Babdi. So what I'm showing you here is an example of what a joint model might look like. Don't worry too much about the details, but the point is, what we're trying to do is we're trying to estimate some properties. Those properties are on the top, including that orientation distribution function. And the way that we do this is we have a model about how those properties we're interested in feed into both data sets, both into the MRI data set and the histology data set. So essentially predict from the estimates that we have, what would microscopy and MRI data look like? Now, of course, those models are different. They have a different relationship to the properties of interest. So, but what we can do is then we can ask the question of how accurately are we predicting those two data sets simultaneously and use that to then loop through and optimize those things that we've estimated. So basically, we have to satisfy agreement with both kinds of data. And that's what gives us confidence that our model is accurate. So an example of what we did with this is a really simple known degeneracy in diffusion MRI, which is that it's difficult to distinguish between whether fibers are disperse or whether you have what we call radial diffusion. It just means that there's a lot of diffusion perpendicular to a fiber tract. If you don't know if you don't know which thing is the thing that is changing, you cannot distinguish between them. And so you have what's called a degeneracy. So on the left, what I'm showing you are a bunch of points which all fit the data equally well if we only have diffusion data in the model. But as soon as we add microscopy data into the model, we get what's on the right. And you see that we can nail it. And that's because the microscopy data has information about what the true dispersion is, whereas diffusion can't tell these two things apart, right? So this joint model is able then to resolve that degeneracy. And if you do that, then you can start to pull things out of the data that you can't find if you're only looking at diffusion. So this, on the left, this constrained spherical deconvolution, this is a way of estimating fiber populations if you only have diffusion data. And on the right, this is using data that also has microscopy. And this is in that centrum semioval region. And you can see that when we have both types of data sets, we're now starting to see that there are actually crossing fibers when we can't find that out from the diffusion data on its own. Okay, but more recently, we've started to get a bit more ambitious in terms of how we use the data. And in particular, I'm going to focus on this data set called the Big Mac data set that, again, Amy Howard finally finished curating and published 2 years ago. And what's really special about this data set is that we have microscopy and diffusion MRI in a macaque brain, in the whole macaque brain. And what Amy was interested in doing, and now this is with a student that Amy and I co-supervise named Tsai Chu, is we wanted to see, can we really leverage the strengths of each method? Oftentimes when you try to combine data, you end up having to deal with the worst of both worlds, right? You end up compromising both data sets. We wanted to see if we could actually leverage the strengths of each data set. And in particular, in microscopy, we have high spatial resolution, so that's great. We're down to, in the case of the PLI data, about 4 microns resolution, but we only have the orientations in 2D. We can't resolve the third dimension of fibers at all. On the other hand, in diffusion MRI, we have pretty poor resolution. So I say 1 millimeter isotropic here. Some of the Big Mac data set is up to 0.6 millimeter, but still very, very crude compared to what we have in microscopy. But the orientations are in 3D. So what can we do with this kind of data to be able to leverage the joint strengths and hopefully ultimately improve the tools that we have at our disposal? So what Tsai did was to come up with methods for estimating, and I don't have a slide on exactly how she did it. Okay, fine. Of building on the strengths of the two data sets. So essentially what she did, sorry I don't have a slide on it, what she was able to do is to take matched voxels and take the fact that in diffusion data we can estimate multiple fiber populations. So with the microscopy data, at its native resolution, you take your in-plane orientation, and then you match it to the fiber in the diffusion data that is the best match to that in-plane orientation, and then you adopt its third dimension. So crucially, you're not just taking a single third dimension orientation from the entire MRI voxel and assigning it everywhere. You're able to essentially super resolve what's happening in your voxel by virtue of the fact that diffusion can find multiple fiber populations, but it doesn't know where they are in the voxel. So you use the microscopy data to tell you where they are in the voxel, right? So in doing that, what Tsai was able to do was to produce results like this, where on the left, I'm showing you this, so what I call hybrid PLI. So this is where we've taken the PLI data and we've managed to give it a super resolved third dimension. So these are the orientations that you would estimate at a relatively crude resolution of 1 millimeter, okay? That's not so helpful. We can do that from diffusion data. But because the microscopy data is much higher resolution, you can start to move to 3D orientations at much higher resolution. And so here this is showing taking 1 millimeter diffusion data and resolving it up to 0.4 millimeter resolution and to 0.1 millimeter resolution. And you can see that within the voxel, you have quite complex structure because you're able to take the strengths of these two data sets and combine them. And so with that, she was able to start thinking about some of the problems that we have in tractography. So this is a classic problem, which is called the gyral bias. So if you have a gyrus in the brain and you look at the connections that come in general, they take a very standard form, which is that they come into the gyrus from the deep white matter, and as they come in, they fan out and connect with the central fibers going to the crowns of the gyrus, and fibers coming in more to the edge of the gyrus, turning sharply and entering on the banks of the gyrus. And the problem in diffusion imaging, where we have relatively poor spatial resolution, is that your voxels would estimate orientations that would look something like this. So when you're firmly in the banks, you would estimate the orientation correctly. When you're firmly in the crown, you estimate it correctly. So now we're talking about in the gray matter. But where you're in the intermediate space, you don't accurately capture the fiber orientations. And so tractography algorithms will tend not to follow these sharp turns. Instead, what they'll do is they will go straight through and show connections as being coming up only preferentially to the crown of the gyrus. And this is a very well-established problem in tractography, and it creates extremely reproducible biases that show you incorrect connectivity patterns. And so what Tsai was able to show was using, oh, and sorry, I didn't manage to label these. That's not so great. So on the left here, this is showing tractography from just the high resolution diffusion data. So this is 0.6 millimeter diffusion data. So quite high resolution, but you can see that it still doesn't really manage to capture that gyral fanning pattern. Whereas with Tsai's hybrid data, which is on the right, you're able to see even in the raw data that you can see this clear pattern of fanning of the fibers, and that the tractography is able to capture that well. So this is another example where she solved, it was looking into what we often refer to as bottleneck regions, so regions where fiber populations kind of have to converge into a relatively narrow tract, and then often will come out the other end and separate again. And so in this case, what she's showing is the ability to take the motor cortex, the 3 different regions, and demonstrate the ability for her improved PLI diffusion data to keep those tracts separate, because they should maintain a consistent separation anatomically as they move through the brain, and in particular as they move here through the posterior limb of the internal capsule. So again, the diffusion data that has gone into this is the same spatial resolution. It's just that it has also been informed by the co-modeling with the polarized light imaging data. But that's not so useful in a sense, because all that can do is demonstrate that if you happen to be so lucky as to have microscopy data alongside your diffusion data, that you can do better, right? Why is that useful? It's not most of the time, because most of us don't have that data. So is there something else that we can do to make that microscopy data, in this case in a macaque, post-mortem, benefit human in vivo diffusion, MRI, tractography analysis? Because that would be useful. That would be something that we could take then and analyze large data sets that capture that variability that we're interested in humans. So can we translate from microscopy data, in our case with the Big Mac data set, in a macaque, all the way through to ex vivo diffusion MRI, to in vivo diffusion MRI, and ultimately into the human brain? And importantly, in this Big Mac data set, we have those first 3 things, all those in a macaque. We have also in vivo diffusion data. So what Tsai did next was to try to attack that challenge. So step 1, and here she's using machine learning now, is to learn the mapping from hybrid PLI, so where we have these improved estimates at very high resolution of what the orientation fibers are doing. Can we learn the mapping from that to conventional diffusion MRI data? And can we do that in a way that's tissue invariant? So importantly, she did this using not just a predictor, but including a discriminator that is assigned the task of guessing, being able to identify whether a given data set is in vivo or postmortem. So when you train networks this way, it means that over time, they will come up with a prediction that is unable to distinguish between data sets. So it's now invariant to whether the data was in vivo or ex vivo. Okay, so all of this was done on a macaque. So now we have something that we can use where we input diffusion data, and it's able to predict the PLI diffusion hybrid estimates. So conventional data, it can predict the improvements that we get when we do have microscopy data. So now you've got something that predicts the microscopy outputs, but without the actual microscopy. Now this can only do that in a macaque brain. So in a second step, she then took in vivo macaque data sets along in vivo human data sets and refined those models such that they cannot distinguish between the species. So now, again, you've created something that is invariant to the difference between the data that you trained it on and the data that you want to apply it on. And so with that, what she was able to demonstrate are results like this. So this is taking that trained network, using it to predict the orientation estimates from diffusion data, and then running tractography. And these are just two examples where you can see that the tractography results, so this CSD tractography, this is just normal analysis of diffusion data, and you can see that it predicts it identifies very chaotic patterns for the corticospinal tract, whereas the network does a much better job of producing clean representations of the corticospinal tract. Similarly, in the optic radiations, it's able to produce a much cleaner version of that tract. So you might be thinking, all of this stuff I've told you about, that's great. I don't have access to this kind of data, except you do. As mentioned, the digital brain bank resource that we have. So this is something that we put out a couple of years ago now, which includes many of the data sets I've been showing you today. We have these sort of three categories: the digital anatomist, so that's that very high resolution data set, as well as a few other similar data sets with some microscopy data alongside them. Digital brain zoo, this is just lots of different species that I'll tell you about in a second. And then digital pathologist, where we have these data sets, in particular in ALS, that are co-registered histology into human whole brains. So the digital brain zoo, this is really fun. I absolutely love this. It's for Here Mars's baby, but he's been scanning every brain that he can get his hands on. It is everything from lots of non-human primates, but also carnivores, cetaceans, all sorts of species. And we even have a Tasmanian devil that Greg Burns was kind enough to put into the database. So that's quite cool. Oh, and the thylacine as well. Okay. Now, we don't just work with ex vivo data. I came from a background of working on pulse sequences and image reconstruction. If I'd arrived the day that I was expecting to, I would have given a lecture to Todd and co. About some of this stuff. But I'll just show you quickly a few images. So this is a 0.5 millimeter diffusion data set in vivo that my student Tsai Li has managed to acquire. It's very slow to acquire this data, so we're going to be trying to figure out how we can make these acquisitions faster. But we have got the SNR, and importantly, as you can see on the left there, we've got very good distortion correction integrated into this so that there's great agreement with the structural anatomy. So that's overlaid on top of the structural. And he's managed to show, again, the ability to get really nice tractography out of this. So another example of this gyral bias, where you can see at 0.5 resolution in vivo, you get this lovely depiction of that fanning architecture, whereas at 1.2, which he seems to think is like conventional resolution because he's been working on high resolution data his whole PhD. For most of us, that would be quite high resolution data, but he's able to show that you're able to do better than the 1.2 millimeter data set. Okay, so with the time I have left, I'm going to quickly go through a different kind of scales. And for some of you, this is maybe more familiar in terms of the work that I do, which is thinking about the scales of people. So imaging of populations. And here, this is taking us, of course, into the UK Biobank data set. So very quickly, I'll go through the aims of it and then tell you about some of the things we've done with the data. So Biobank is a massive epidemiological study. The aim is to acquire huge amounts of data, deep phenotyping of a large number of individuals, lifestyle, blood chemistry, genetics, and imaging, alongside the long-term health outcomes. So we have access to, via the National Health Service, we have access to the health outcomes of all of these individuals, so they consent to have their data shared with researchers, so that you can learn relationships between these things with the ultimate aim of being able then to predict long-term health outcomes in new individuals from outside of the study based on the kind of data that you can collect in a fairly normal clinical setting. So I love this video. I'm going to show you another video. This is a video comparing the Biobank to what has come before. So if we start with a relatively generous interpretation of what is typical within the imaging community, if we say it's 125, this was pretty much what the biggest studies were when we started the Biobank at 1,000. This, then, is what the UK Biobank looks like. It's 100,000 people. It just dwarfs anything that came before it. And the idea is that because we're scanning people before they're sick, you can find a needle in a haystack. So you can find, for example, the prediction that 50 people are going to go on to get ALS within the first 5 years, 2,000 people will get Alzheimer's diagnoses. So the only way that you can get at this information without having any ability to predict already who is going to get these diseases is to just scan everyone. And statistically, you will cover people who are going to ultimately go on to get these diseases. So just very briefly, this is part of a broader study of 500,000 people. It's been going on for quite a while. They're extensively characterized. I've said most of these things. And we're trying to come up with reliable assessments of many causes of many diseases. So there's no disease of focus in particular. So with the imaging study, then, what we're doing is we're bringing back 100,000 of those individuals. And as you can see on the right, we're not just getting brain imaging data, we're getting body imaging data, we're getting carotid ultrasounds and bone scans. And really importantly, they're all scanned prior to, well, I shouldn't say that. Most of them are scanned prior to major health outcomes. We don't screen people out if they do have major health outcomes, but based on their age, they are broadly healthy at the time of scanning. And again, no disease of focus in particular. We're just casting a net and playing a numbers game. So we're limited in the time that we have. We have 35 minutes. But we managed, after a lot of protocol development in that time, to get in most of the scans that we would include in a multimodal research study. So these are the things that we decided to focus on. So we have structural scans, T1 and T2 flares. We have diffusion data. We have resting as well as a single task for functional MRI and susceptibility-weighted scans. So quite a rich data set that we managed to shoehorn into that short time frame that we have. So I'll just give you a feel for some of the results that have come out of this. So one of the first things that we did was to compare the imaging data to non-imaging phenotypes. So I'll show you these plots before. If you haven't seen these are called Manhattan plots. Vertical axis is negative log 10p, so the higher they are, the more statistically significant. And this is just taking 5,000 imaging phenotypes that we derived from those 6 modalities that I showed you, and doing pairwise associations with 1,100 non-imaging phenotypes, things that were already in the Biobank database. And what this is showing, so we did 2.8 million association tests, and FDR level of multiple comparison correction, we identified 30,000 significant associations. So the main point of this is to give you a feel for what happens when you start to get to large numbers. Now, this is a large number of subjects. It's 5,000, but it's only 5% of the ultimate data set, right? But it gives you a feel for the power of the data set. We can also talk about what on earth one does when one has 30,000 pairwise significant associations. This is an interesting topic of discussion, but this is just mainly aiming to give you an idea for what happens when you start to have numbers this large. It also provides opportunities to do things we haven't really been able to do before. For example, genome-wide association studies. So if you haven't come across GWAS before. It's basically the same thing that I was showing you before, lots of pairwise associations. But now instead of the pairwise associations being with non-imaging phenotypes, they're with loci along the genome. So you go along, and at every locus in the genome where there's a known variation between individuals, you do a correlation. So to give you an example of what that looks like from the first paper that we had, so this is an example of the same kind of plot, but now with the vertical axis indicating statistical significance, but now it's the horizontal axis is where you are in the genome. In this case, it was based on using an imaging estimate of T2 star in the putamen, so one particular quantitative measure in a deep gray structure. So if you take that median T2 star in the putamen and you correlate it across the genome, you find about 4 different spatial or 4 different loci or SNPs that are statistically significant. Now, what's interesting, I mean, that's interesting in and of itself to try and understand what are those genes telling us. But an interesting thing that you can do with imaging data is you can then take those peaks, those peak loci, and you can use them to regress back into the imaging data itself. So to say where in the brain at a voxel-wise level are there associations across individuals with this particular locus. And what's interesting is if you then look at the spatial maps that emerge, they all include the putamen. We knew they would because that's how we found them in the first place. But they also segment deep gray structures in fundamentally distinct ways. And so this is telling you that they're not all giving the same information. They are all associated with these genetic loci in unique ways that involve different deep gray regions. And interestingly, if we look, for example, at one of these, and I believe this is the pink one that I'm showing on the bottom there, you can also start to interrogate what that association might mean. So in this case, this particular locus is related to a gene involved in iron transport and storage. And that's really compelling when you think about what T2 star is, because it is an imaging marker for tissue iron. Okay, I'm going to give you 2 more quick examples, and then I'm going to stop so we have time for questions. So I know that there's some interest here in brain age modeling. So I'll just talk a little bit about one thing that we've done with brain age modeling using the Biobank data. So brain age modeling, very crudely, what one does is you take imaging-derived phenotypes. So these are just 3 examples of phenotypes that you can extract from brain imaging data, and you use it to form an age prediction. So can I predict the person's age simply based on imaging features? And so you can see here that we can form a perfectly good age prediction. Of course, we know the person's age, so why would we do this? Well, we do this because we're not interested in the brain age prediction. We're interested in how wrong we get it. So if in an individual we get their brain age prediction very wrong, like this individual, we can calculate how wrong we got it, which we often refer to as the delta, or the brain age delta. And the idea is that a positive brain age delta, it indicates that their brain is aging faster than the population norm. Now, I'm going to put an asterisk under that. Is it aging faster, or was it already offset at birth? This is a different point that I think Steve probably talked about in his talk this morning. But the idea is that your brain looks abnormally old or abnormally young, and that might be biologically interesting. So we didn't come up with this concept. Many people have done it. This paper is just our first foray into it and trying to understand how to accurately come up with these estimates. What I was particularly interested in was the question of, should we be thinking about this in terms of a single delta? Does it make sense for there to be a single brain age delta? We all know that people age in different ways. You could be very youthful in one sense and very old in another sense, depending on what sort of a biological cause you're interested in thinking about. So we don't really expect there to be a meaningful single brain age, but rather that we are all on different aging trajectories according to different metabolic or lifestyle choices or genetic predispositions. So one of the things that was really powerful then about having so many subjects in the UK Biobank data is that it does allow you to start to do more sophisticated things. And so in this case, what we were doing was trying to identify multiple modes of brain aging. And we did this in a sort of semi-supervised way, identifying modes of variation in the population and asking, do they correlate with age? So we did this, if you're familiar with ICA. This is an ICA-based approach. And we simply discovered modes of variation in an aging population, looked to see which of those were actually correlated with age. And most of them had quite a strong correlation. So you can see here, like the strength of the correlation across a large number of different modes. We can go into what some of these modes seem to correspond to, but rather than that, I'll skip forward to the thing that I found to be really convincing about whether these different modes and their association with age might actually be meaningful in terms of different biological mechanisms of aging. And that was this. So this is showing genetic associations with the brain age deltas from each of these different modes. So each mode is in a different color, and sometimes in a different symbol. And what you can see is that, and on the top, where you keep each of these deltas, you look at them separately, so each individual mode has its own specific pattern of genetic associations. And that, to me, is strongly suggestive that it is indicative of different biological processes relating to age. But you can only really discover this when you have data that's rich enough to drive these kind of data-driven estimates of brain age deltas with high dimensionality. If you compare that to what you do if you calculate a single brain age delta, the genetics of that is on the bottom. Everything falls away. You have no genetic associations. And the reason for that, I think, is that what you've done is you've gotten a better age prediction, but you've diluted out the different, in doing that and creating a better age prediction, you've diluted out the unique biological content of these different modes. So you've maybe said, well, it looks like you may have Alzheimer's or you may have high blood pressure. You're probably old. But it's combined those two things in a way that has lost the biological meaning. Okay, so with that, I am going to, actually, I'm just going to mostly end there. I'm going to go to this slide that acknowledges all the people in Biobank. It's a big project. We tend to be, getting to stand in front of people and tell you, but it is a lot of people involved. I hope you see some friends in there. They're probably in there somewhere. And then I'd like to thank my group, and thank you for listening.